All right, so in this video, we'll continue with Unit 1 of our basics of anatomy, and we'll be looking at cell basics and then the cell membrane. A lot of this is going to be review. More than likely, you've had this at some point. If you've had honors biology, for instance, I know you've had this, and so this will be review, but it doesn't mean it's bad. It just means make sure we're on the same page. All right, so cells, a couple things about cells. They're the basic unit of life. They're the basic unit of structure and function. Larger structures function is based on the cumulative function of individual cells. So what does that mean? All right, so like a stomach is made up of cells. The stomach's function has this digestive function, has many facets, but it can't do that without the function of the cumulative effects of all the cells that are in it adding together. And so make sure that you understand that. Cells have three main parts. They have a plasma membrane, they have cytoplasm, and they have a nucleus. We're going to be going through those parts in the next couple of videos. You see a couple of different kinds of cells there on the right that we are going to talk about throughout the days in this unit and in upcoming units as well. So there is some fluid that is outside of the cell. It is called extracellular fluid. Here's a picture of a rat brain, and this gives you kind of a picture of that extracellular space. When you think of an organ like a brain or whatever, it's not completely jam-packed full of cells. There are cells in there, but there's a lot of space that is not cells as well. Some parts of the body have more of this extracellular space than others. A lot of this space is filled with this extracellular fluid, which includes could be blood plasma, it could be interstitial fluid, which is just kind of this in between the organs kind of fluid. It could be cerebrospinal fluid, which we'll talk about in the nervous system unit. The purpose of this fluid is just to carry nutrients, to carry waste. Could have hormones in it. It could have enzymes as well. There's a lot of things that are going on in this extracellular space and that don't necessarily involve cells directly, or they're headed to a cell to do some sort of work. And that's a lot of it just has to do with keeping the cells in place as well. There's this structure called the extracellular matrix, and the purpose of the extracellular matrix is it's all these proteins and carbohydrates that hold the cells together. They allow the cells to communicate between each other. You can kind of see all the different things that are going on there. Feel free to look each one of those up individually and learn about them, but you don't need to do that. You can just do that on your own time. There's a lot of big words there. Have fun with that. All right, so let's talk about the plasma membrane. Plasma membrane is composed of molecules called phospholipids, which you can see they're very small there. And we've had this before. Phospholipids are these hydrophilic heads and nonpolar hydrophobic tails. They kind of look like little dudes with a head and no body, just legs. Or I don't know what that looks like to you. That's what I think about. All right, and so you can see the other things that the phospholipid bilayer is made out of. It's not just these phospholipids, but there's other things going on as well. You can see there's cholesterol there, and it shows the cholesterol. There's proteins, we'll talk about those. There's carbohydrates as well. And so this plasma membrane, sometimes I'll call it phospholipid bilayer, sometimes I'll call it cell membrane. It's all basically referring to the same thing. They are all working together to keep the cell separate from its outside world. We've talked about homeostasis. The cell is also attempting to maintain an internal environment that is different from the outside. So different cells are going to have different kinds of internal environments, and that's important. There are different kinds of proteins that are located on the cell membrane. You can see them here in this picture as well, but they have names. They are the integral proteins here, and the integral proteins go all the way through the membrane. You can see here, there's the membrane, there's the maybe inside of the cell, and here's the outside of the cell, and that protein goes all the way through the membrane, whereas a peripheral protein doesn't go all the way through. It's on the periphery, on the outside of that cell membrane. And then there's another structure called the glycocalyx, And the glycocalyx is this carbohydrate covering that aids in cell recognition. So you have these proteins, some of them are peripheral, some of them are integral, and you have these carbohydrates that are kind of sticking out here on these proteins. And the purpose of those carbohydrates are so when cells are rubbing up against one another, they recognize each other. They're like, oh, yeah, that's one of our cells. Or, oh, no, that's not one of our cells. We need to do something about it. And so that's the purpose of that glycocalyx. Next are this concept of cellular junctions. All right, what is a junction? It's where two things join together, right? And so most cells are bound together through these glycoproteins, which is a glycoprotein is just a sugar protein combo with these interlocking kind of regions that are joined together in one way or another. Some of them are really tight, some of them not so much. And so we're going to look at three different kinds of cellular junctions, and you see them down at the bottom. There are tight junctions, and these tight junctions, think of them as like stitching between two cells. All right, they prevent the passage of material between them. So it's just like if you had, if you had stitching between two pieces of cloth, you couldn't like put your hand through that stitching. It prevents the passage. A very tight fit. Whereas the desmosomes here, these are like anchoring junctions. Think of them as like rivets. If you're trying to put two pieces of metal together and hold them together, you use something called a rivet, and it holds those two pieces of metal together. It kind of anchors them together. This prevents tearing between those two cells. Think about your skin. If you just pull the skin on the back of your hand, it all stays together, thankfully. It's because of these desmosomes. They give that skin some real structure. And then there's gap junctions. Gap junctions are different because they allow passage of material between two cells. And you can see that in that picture, right? It's like a little tube that passes between the two cells. It's kind of like a tunnel between two rooms, if you want to think of it like that, like my room and Mr. Krug's room. There's not a tunnel. It's like a little room there. It's a closet. It'd be fun if it was a tunnel, but it's not. But you get the idea. It's a way that those two cells can like talk to each other or pass materials from one to the next. And next, we're going to look at cellular transport. This is a deep rabbit hole that we're not going to go deep into. In other classes, I do go deeper into this, but we're going to just divide it into two main things, passive and active transport. And I'm going to give you a couple of examples of passive transport and nothing for active transport. So passive transport is the movement of materials without using energy. It just uses a concentration gradient to go from an area of high concentration to an area of low concentration, kind of like rolling a ball down the hill. If you put a ball on the top of a hill, the ball does not sit there. It rolls down naturally. You don't even have to push it because of that concentration gradient, or in the case of a hill, it's an elevation gradient, but you get the idea. If you put a drop of blue food coloring in a glass of water, that food coloring will disperse throughout the entire glass. It's because it's moving from an area of really high concentration that was once inside that drop, and it's spreading out to the lower concentrations until everything is equal or at equilibrium. So there are some basic kinds of passive transport. There's simple diffusion, which is kind of what we just talked about with the glass of water. Simple fusion. Diffusion can happen across a cell membrane as you see these materials passing straight through the cell membrane. There is facilitated diffusion, which is diffusion using some sort of channel like you see here. You don't need to know the difference between the two here, but just facilitated is using some sort of channel. And then there's osmosis, which osmosis is just the diffusion of water. And remember these, you probably remember these words like hypertonic and hypotonic. Those words just have to do with the amount of solute. And so if you want to look here, that if you want to just use solute here, and solute is this yellow stuff, not these actual water molecules, the inside of the cell has more yellow than the outside. So the inside would be hypertonic and the outside would be hypotonic. It's like if you put a raisin in a bowl of water, the inside of the raisin is hypo or hypertonic because there's more raisin in there than there is on the outside. And water typically goes into the hypertonic. Some water will move from hypo to hypertonic. Not going to spend a whole lot of time there. It's more important in some other classes. So for our purposes, just make sure you understand passive transport, no energy. Active transport, on the other hand, does require energy. This is like pushing the ball up the hill. It will not go on its own. You actually have to put energy. And so if you look here at the top, these little purple molecules are able to go into the inside because there's more purple on the outside than there are on the inside. In this case, the purple is being pushed inside the cell. There's more purple inside this, or more green inside the cell. There's only 2 out here, but those 2 are going to be pushed into the inside. It's kind of like loading up a crowded closet and then slamming the door. Now, if you were to just twist the knob on that door, what's going to happen? All of that energy that is stored up inside the closet is going to release, and it's all going to fall down on top of you. And so in much the same way, the cell can use that energy that is stored up from active transport in order to do things. There are a lot of cellular processes that require these concentration gradients in order to do work.